Okay guys, so here we go. We are on our last chapter for the year, and then we will start reviewing after we have our last unit test. So far we've gone over most of evolution, but we haven't discussed early Earth. In other words, we haven't discussed how life actually started. And that's what we are going to go over today. In the next few days, we'll talk about the hypotheses and the theories and all that type of stuff. So we know that Earth formed about 4.6 billion years ago. However, it took life about a billion years to start after Earth was already formed. So you can see there, 4.6 billion years ago, Earth was formed. We didn't have our first prokaryotes until 3.5 billion years ago. And these first prokaryotes, they were simple. These are things like bacteria, which you guys know bacteria, and archaea. We've talked about that in the past. These are our prokaryotic organisms that live in hot springs and other extreme places like that. So here is a picture of what we would think early Earth would look like. Now, when we talk about prokaryotes, we are talking about a very ancient organism. And there's a whole bunch of different ones. And if we talked about bacteria, there are so many different species of bacteria on Earth that it would well overpower any of our number of animal species that we have in existence. So I know we can't see prokaryotes without microscopes, but just so you guys know, they are much more plentiful than any organisms that you can see. So kind of a side note, and this is kind of interesting. We have honey here, all right? And I know you guys have probably had, honey before, but along with honey, there's other things that are really, really sweet, like preserves, jams, things like that, jelly. So when we're talking about prokaryotic cells like bacteria, the cool thing is with honey, it actually doesn't have a real expiration date. Now you might go to the store and it might say, oh, this honey expires on this day. Yeah, maybe it's not pure honey, or maybe they put something in it. But if we have pure honey that's just honey, bacteria actually cannot live in it. Reason being is the honey is so sugary and sweet that it will actually suck the water right out of the bacteria, causing the bacteria or any other prokaryote to actually die. So that sugar concentration is so high. In other words, the bacteria, if we went over osmosis, the bacteria is put into such a hypertonic environment that it sucks the water right out of the bacteria and it can't live in there. So honey actually really doesn't have a shelf life. And here is a picture of a person putting their hand on something, and then they let bacteria grow where the hand was. You guys can see that's where the bacteria was present. So we do have bacteria all over our bodies, and it's also in our bodies as well. It's not only on your skin, but it's in your stomach, it's in your intestines, all these different places you have bacteria, and most of it is good bacteria. That bacteria is performing some kind of duty in our bodies. Okay, so we're going to start the chapter now, guys. That was just a little introduction. We're going to go over some of the first cells and how we think they came into existence. So I'm just going to go over on this slide protocells. So these are like the prototype cells. In other words, it was pretty much like droplets that they had some kind of chemical composition to them, and we think that these were the earliest parts of the cells we know today. Now, early Earth contained not as many gases as we have today. Mostly, the main gases on Earth were nitrogen, carbon dioxide, methane, ammonia, and hydrogen. Now, if you're looking at what is in early Earth with respect to our gases, we're missing a very important one, and that's oxygen. In early Earth, there was no oxygen, which is really interesting. So how did we get oxygen? Well, some of these early organisms started to photosynthesize, and when they started to photosynthesize, we started to increase our levels of oxygen on Earth. And that took a very long time. But this was pretty much all we had in early Earth. So what they were trying to do here, guys, we had a few different scientists, and they were trying to come up with how these gases, the ones that we had in early Earth, started to form things that were able to form our cells. So in other words, guys, we have gases, but those gases, it's not going to be like, oh, I'm made of methane or I'm made of carbon dioxide. No, we're made of nucleic acids. We're made of carbohydrates. Okay, we use carbohydrates for energy. We're made of proteins, amino acids. All right, we're made of fats, lipids. Okay, how did these gases correspond to our macromolecules in pretty much giving us these ingredients that we need for life? So first scientists here, Oparin and Haldane, they hypothesized that Earth's atmosphere was a reducing environment. So what we're saying here, guys, reducing, go back to, Leo the lion goes grr, lose electrons, oxidize, Leo, gain electrons, reduce. So we were removing and changing these electrons so that we can get new combinations of elements with respect to using those early gases, the methane, the carbon dioxide, the nitrogen, the ammonia. So what we were trying to do is they were trying to figure out a way to form amino acids. So Oparin and Haldane, they had the hypothesis. And then the next scientists, a few decades after this, Miller and Urey, they actually conducted the experiments so that they could prove that these gases were able to be used to make amino acids, so parts of a protein. I'll post a video for you guys on their experiment. But what they did is Miller and Urey, they used electric shocks to try to recombine the elements of our early gases on Earth. And in recombining these, they were able to, with their experiment, they were able to make amino acids. Now they realized that when they were doing this, you can't just have these gases and it's like, okay, hold on, I'm going to take a carbon from here, okay, and we're going to take a nitrogen from over here. It doesn't work like that, okay? These things are much too small and they are chemically bonded together, so we can't do that. So what they had to do is try to think, okay, hey, what was on early Earth? We had volcanoes. We had an atmosphere. We were getting an ozone layer and things like that. That was forming. So what did we have that we can use to reorganize these elements into amino acids, into the monomers of proteins? So what they did here, guys, is they actually used lightning. Now in their experiment, they didn't, okay, hold on, let's wait for the weather to do its thing. They used electric shocks that would simulate lightning strikes. And that's what they did in their experiment. And in doing so, they were able to use the lightning, and it did reorganize the elements of early Earth to make amino acids. So is it correct? Probably. This is the best theory we have today on how amino acids and some of our other macromolecules came to be. All right, all these different organic compounds. So this is what we think that happened in order for these organic molecules to form. We also found something else. This is part of a meteorite from 1970. This is called the Murchison meteorite, and it came obviously into our ozone. And the funny thing is, when it landed, they actually found amino acids on it. So there's your UFO conspiracy theory, maybe, that there's amino acids not only on Earth, but there's some in space. Does that mean there's life out there? Nobody knows. All right. What we're finding now, guys, is if you look at these two charts, these are massive amino acids. Now, we're finding that our number of amino acids is going up. We are actually having more amino acids as time goes on, and furthermore, the mass of those amino acids is increasing. This tells us that our atmosphere is changing. Not only is it changing, but it's also still able to manipulate some of our gases and make them into amino acids. Okay, so now we're going to get into RNA. So RNA, if you guys remember, that is one of our nucleic acid macromolecules. We had RNA, we had DNA, ATP was also one. But we found that some of these organic molecules like proteins and stuff like that, they were really concentrated on hot sand, clay, and rock. And what we did was we started to get these protocells. So the protocells, guys, were pretty much like just little vesicles, and the vesicles just had a membrane structure around them, and that was pretty much it. So we're going to use these, and what they did was they took the protocells and they added clay to it, and they found that the vesicles, these protocells, were starting to increase in formation when they had some kind of medium, like clay, to reproduce in. So things were going faster. What we were finding, guys, is the RNA was also sticking to the clay and was able to actually move into the vesicles, the protocells. So now we got these protocells, which are essentially a cell membrane, and now we got the RNA sticking to this clay, and the RNA was able to get into these protocells. And here's just a graph, guys, of, you guys can see if those protocells are in clay up top or are not in clay down below. Okay. So you guys can see here it says relative turbidity. Turbidity is just like the cloudiness or haziness of a fluid, and this is caused by the number of particles that are inside of that fluid. So the more cloudy, the more hazy it is, the more particles we have inside. So that's how they measured that data. What we found, guys, was that the first genetic material was RNA, not DNA. And your book there says probably. I'm going to tell you it's definitely. Reason being is we would not have DNA without RNA. If you remember when we went over DNA replication, we needed RNA in order to start DNA polymerase, to start adding those nucleotides onto the new growing strand of DNA. So without RNA, we would never have DNA. So RNA definitely came first. This also means that since, remember, RNA has uracil and DNA has thymine, this also tells us that uracil actually came first before thymine, and thymine was probably a mutation of the RNA nucleotide uracil. Okay, so then we're going to talk about ribozymes just a little bit, guys. These ribozymes, I know we went over them in the past, they act as protein catalysts or enzyme catalysts. So they are able to actually be the enzyme that is going to connect these amino acids together to form a protein. So they're able to pretty much mediate amino acid binding so that we can make a protein. All right. Okay, so we found that when we were getting these protocells and the RNA was slowly getting inside of it, found that these RNA molecules were actually pretty stable, and they were able to replicate very quickly whenever they were inside the protocells. So in other words, we had one cell with RNA inside of it. It was able to self-replicate to form another stable cell with RNA in it. So we think that it was mostly an RNA world in early Earth. So we already talked about the second one. RNA was the template for DNA, and the DNA was actually even more stable than the RNA. Now, why was it more stable? Well, if you remember when we went over polymerases. The DNA polymerase had that little spell checker on the back to make sure that there was no mutations in our DNA. RNA polymerase doesn't have a spell checker, so we have a higher probability of mutation in RNA compared to DNA. So what scientists are doing now is they are trying to find out if the origin of life on Earth does deal with RNA. In other words, was RNA the first one? We know RNA came before DNA, but was there something before RNA? We're also trying to wonder how RNA came to translate. You guys remember when I say translate, I'm talking about translation, RNA to a protein. They're trying to find out how that RNA came to translate for those amino acids. Right now, scientists really aren't able to determine how that happened currently. We know that the mRNA binds to the tRNA and the tRNA pulls that amino acid over, but how did all those correspond to one another and how did all that start? Okay, so when we look at the fossils of early Earth, we find these stromatolites, and we talked about stromatolites last chapter, and they're like almost rock formations, and these rock formations were held together into different parts by prokaryotic cells. Now, when we found these stromatolites and broke them apart, and we found the prokaryotic cells, obviously using a microscope, we found that they were approximately 3.5 billion years old. All right. Now, we're not actually looking at the prokaryotes. We're looking at the rocks and finding how old those rocks are that were fused together. And here's a picture, guys. You can see there's all the fused plates of the stromatolites there. So these prokaryotes that form our stromatolites, that connect them together, they're called cyanobacteria. Now, cyanobacteria is a prokaryotic photosynthetic organism. So cyanobacteria, it's a prokaryotic cell and it's able to go through photosynthesis. This tells us that cyanobacteria was our first organisms that began to release oxygen into the Earth's atmosphere. So we were switching from pretty much having no oxygen in Earth's atmosphere to slowly, very, very slowly, having these levels of oxygen increase. Now, when the oxygen levels increased in early Earth, some organisms were fine. They're like, hey, oxygen, okay, no big deal. I don't want it, but it could stay here. Other organisms actually couldn't handle the oxygen. Since they couldn't handle it, they actually died, and that was our first big extinction. All right, so our first mass extinction was because of oxygen entering the environment, and a lot of organisms couldn't adapt to that, so they died. Okay, so we talked about in the past prokaryotic and eukaryotic cells. Prokaryotic cells, no nucleus, no organelles except for ribosomes, and eukaryotic cells, they have a nucleus, they have organelles, all right, and they are much bigger. You guys can see how much bigger they are between the micrometers difference between our prokaryotic cells here and our eukaryotic cells down here. Now, when we're talking about bacteria, bacteria can have three different shapes. Now, the three names are on this slide right here, cocci, bacilli, and spirilla. Let me go right to the diagrams of them. Cocci, this is a shape of bacteria that is circular. So that's cocci, C-O-C-C-I. Bacilli, it kind of looks like Cheetos, almost like little rod-shaped things. So, or cheese puffs, we could even say. But this is bacilli, B-A-C-I-L-L-I in the middle here. You guys can see it's not circular, it's more of a rod shape. And our last one kind of looks like spiral pasta. This is called spirilla, all right, another shape of bacteria. So when you guys have bacteria on your skin or have bacteria in your body, or thirdly, get bad bacteria, they are all going to be in one of these shapes here. Now, when we're talking about bacteria, they are going to have a cell wall. Now, us animals, we do not have a cell wall, but plants, they do. Some other organisms, they do, but we do not. Now, bacterial cell walls are made up of a protein called peptoglycan, and you guys can see that there. Now, the eukaryotic plant cell, it's going to be made out of something different. We are either going to have cellulose or chitin. Now, I'm just going to talk about cellulose for a minute here. If you guys remember from the past, cellulose for humans is unable to be digested. So our bodies can take all the nutrients from inside, but we actually can't break apart that cellulose to use it for too much. So it pretty much just goes to waste for us. Okay. So we'll be finishing up here soon, guys. Bacteria, depending on what type of cell wall it has, some of them can have a lot of peptoglycan in it. Some of them can have very little peptoglycan in it. And if it has a little bit, the outer membrane can actually be toxic. Now, what we can do is we can do something called a gram stain. We will stain the bacteria to see if it is gram positive, meaning it has a large amount, or gram negative, which means it has just a little amount of peptoglycan in it. Now, I can't see these very well, guys, because I'm actually colorblind, but I do know that, I did this lab in college, we have gram positive bacteria, and when we stain it, it will become purple. So the cell wall will actually show purple, meaning it has a large amount. If it is gram negative, we will have a pink color in it, and that will show that we have not so much peptoglycan. Okay, and you guys can see the difference here between our gram positive one over here with respect to the cell wall and our gram negative one over here with respect to our cell wall. And you can see this, the gram negative one, we have the peptoglycan layer sandwiched in between the cell membrane down here and another outer layer which is completely different, and it is a selectively permeable membrane as well. So what many antibiotics do, guys, is they actually target that peptoglycan, because our cells in our body, we don't have a cell wall, and we're not going to have a whole lot of peptoglycan we're not going to have any peptoglycan in our cells. But gram-negative bacteria are more likely to be antibiotic resistant because of that low number of peptoglycan. Okay. All right, so other things with bacteria, guys, I wanted to go over. And I know we've seen the prokaryotic cell in the past. These things out here, let me go back to our slide here. So there's going to be tiny projections on the outside of our bacterial cell on that outer layer called the capsule. So I'm mainly going to talk about pili. All you have to know for the first one, guys, the fimbria, all this does is help the bacteria to stick to things. All right. The pili, these are going to be longer than the fimbria, and this is going to allow, because it's already going to be stuck to something from the fimbria, the pili is going to allow that bacteria to exchange DNA with other bacteria. Now let's talk about this, why this is important for us. And then after this, we'll be done for the day. If we have our bacteria able to exchange DNA with other bacteria, what that does is if we have a bacteria that survived and it's antibiotic resistant, it will exchange that DNA. It'll give that code to another bacteria, and that bacteria could potentially become antibiotic resistant as well, or at least know what it needs to do in order to be antibiotic resistant. So the pili, does help an exchange of genetic material between bacterial cells. This is making it become pretty much sexual reproduction. All right, guys, so we will stop here for the day. Have a good rest of your day, and we will continue our last chapter tomorrow.